So it's my great pleasure to introduce Monica Fabiani. She's a professor of psychology in the Cognitive Neuroscience Division at the University of Illinois, Urbana-Champaign, which I went to. And she was on my dissertation committee, so. And today she's going to be talking about, what's the?
It's the next two slides.
Mens sana in corpore sano. A sound mind in a sound body, aging, fitness, and cognitive health. So I'd like to welcome her.
Thank you. It's really a pleasure to be here. I love coming to Berkeley, also because my daughter is at Berkeley. And this has been an absolutely wonderful conference, a lot of food for thought for future studies. So mens sana in corpore sano is an expression in Latin, so the Romans knew it already, that you need a healthy, to have a healthy mind, you need a healthy body, and that's basically the gist of my talk today. I hate this slide, but I am bound to present it because I'm kind of where the two curves cross, and that's upsetting. But basically, this is, in a nutshell, what happens in cognitive aging. Let me see if I can do this. Yes, I can use this pointer. OK, so what happens is that many of the tasks that are sort of online tasks, these are data from Denise Park's lab. Many of the tasks that are online, like working memory tasks that require sort of speed, tend to decline, and this is ages from 20 to 80 if you're in the back and I cannot read it, while other tasks like vocabulary tasks that require acquired knowledge over culture and education and so on tend to grow even on the lifespan. They don't, or at least flatten out and don't decline as much. And one of the points, though, that this graph does not make is the fact that there is a differential variability, so that if you take younger adults, the variability is lower than when you take older adults. So to some extent, understanding aging is also understanding what drives this variability, so that some people age with health and in a state of cognition that is very maintained, and others don't. In the same frame of, in the same framework, you also notice that there are changes in brain aging. There are changes in the brain that mirror sort of some of the cognitive changes. These are now structural MR images, not functional. So the color here indicates areas in which younger and older adults differ, and the younger adults here are college age, the older adults are 65 and older. So in the top row, let me do this because it's probably easier. In the top row, what you see is that in the gray matter, in the cortex, there is loss in frontoparietal network, which sustain tasks like working memory tasks and so on, as well as medial temporal areas that sustain long-term memory. In these same areas, you have an increased cerebrospinal fluid in the older adults, because that takes place of the shrunk tissue, and there is also some loss in white matter, where younger adults have more white matter than older adults. But even, this is the bad news case, but when you look at individual differences, these are data from our lab by Brian Gordon, and these are data from Naftali Ra's lab, and these show you each line is an individual, and now it's still, the regression line is still a decline, but you can see individual people being really high up here, even at a very old age. So this highlights the idea that even in the brain, there are very large individual differences in terms of how much a tissue is preserved during aging. These are more recent data from our lab, from Mark Fletcher, that also highlight thinning of the cortical mantle in the same way. And again, these are comparing younger and older adults. So this is kind of the bad news part of the story, but the individual difference kind of might lead us to figuring out which factors actually one can harness to improve this sort of bad ending story. And so what are these factors that promote healthy aging? And one of the ideas that have been around for a long time was proposed by, can you still hear me in the back? I tend to move when I talk, and I'm worried about fading out. Just let me know if I do. And also, please let me know if you have questions during the talk. I'll be happy to take them when they come and not wait to the end. So this is data from Jaakko Stern's lab. This is from a recent Trends in Cognitive Science paper, showing that if you look at cognitive status, you might identify people that have high and low reserve, where reserve is factors such as socioeconomic status or education that might protect you from or might delay the time at which the cognition leads to sort of an incident dementia effect. And so the idea here is that a person with higher reserve that has more alternatives, more things to do in cognition might hit this line later than a person with low reserve. Of course, this is a simplified version. The version that I think is important here is one that takes into account several things. The first is that there may be changes that don't occur so sharply. So this is a transition point, and this is part of what this conference is about, the idea that there are lifespan transitions, but there are things that continuously change in a quantitative manner throughout the lifespan. And those might be those factors that might be more amenable to intervention because they're gradual. The other aspect of this is that in aging, more than probably any time in lifespan development, one needs to remember that there are many things changing, and not just the brain and not just the mind or cognition. Because there are many organ systems, and if you look at the trajectories of change in these organ systems, they do not change all at the same rate. But for sure, they will interact with what happens in the brain in a significant manner. And so this is one point that I kind of carry along in the talk that I'll give you today. The other point is that, again, the cognitive reserve and the idea of brain reserve, which is sort of the brain counterpart of this, are in fact highly correlated. This is a recent paper by Satz et al. And my research has been very much into sort of trying to find the connections between these two, trying to go full circle, looking at working memory changes or executive function changes in the context of how anatomy or functional activation change, and putting them into one framework because of these reasons here. What I will do today is talk about individual difference in one particular factor, which I chose because it's one where we have some control on. So if you have more brain volume or less brain volume, you can't do anything about it. But cardiorespiratory fitness, the idea of exercise, as the title suggested, as a good thing, is something that, in fact, can be intervened upon in terms of behavioral changes, and so I chose it for today's talk. Now, these are new data. A lot of them are unpublished, so you are actually the first audience I give this talk to. So if something isn't clear, since I haven't had a previous run-through, please feel free to raise your hand and ask, okay? So just to explain why aging and fitness are important, let me give you a little bit of a background. So blood flow and tissue perfusion to the brain decrease with aging due to a number of reasons, including hypertension and arteriosclerosis and plaque formation in the blood vessels. So this is age, and this is cerebral blood flow, and you can see that there is a diminution over age. Now, cardiorespiratory fitness can, in fact, counteract some of these effects by inducing a number of different things. Increase in tropic factors, which is like brain-derived neurotropic factors, BDNF, spinal dendrite, and neuronal growth. These, for example, are data from Gage's lab showing that, this is in mice, that young adult mice, these are stem cells in the dentate gyrus of the hippocampus that give rise to new neurons. It's a schematic. And you can see the aged mice that are that have been intervened upon. So they have an enriched environment in which one of the things that happens is that they move around a lot more, have many more stem cells than the mice that instead are caged individually as it's customary. There is angiogenesis, that it's one of the effects of increased cerebrospinal cardiorespiratory fitness. And in humans, there is increased arterial elasticity. And in humans, there is a lot of work done by our Kramer's lab that shows improve and preserve cognitive function, both cross-sectionally but also longitudinally, and preserve brain tissue, and this is data from our lab as well as our Kramer's lab, in people that have higher fitness or in people who undergo fitness training. So this is both, again, intervention data and cross-sectional data. So continuing with this, there have been an enormous amount of data recently indicating that vascular health is an important mediator of the beneficial effect of cardiorespiratory fitness on both anatomy and function. For example, there are models that suggest that Alzheimer's disease and vascular health are directly linked, so that if in a hyperperfusion, in a case in which the blood vessels don't bring enough juice, this is the technical term, to the neurons, then you have an increased accumulation of plaques. So they postulate a direct link between perfusion and Alzheimer's, and there are a lot of data suggesting that at least the manifestation of Alzheimer's disease is certainly worsened by the presence of, again, hypertension and hyperperfusion and so on. So arterial elasticity, which is one of the topics that I'm going to talk about today, is typically measured in various ways, none of which is ideal to assess what's happening really to the arteries within the brain. So it's measured at the carotids, which are the two arteries that run around the neck. It's measured intracranially by big vessel Doppler, but as the word suggests, this measures only the very big blood vessels and not the arterioles that actually perfuse our entire brain. Or more typically, it's estimated by looking at the difference between systolic and diastolic blood pressure, which is called also the pulse pressure. So if you see here, this is age, and this is blood pressure. This is the systolic blood pressure, the high blood pressure, and the low blood pressure, so to speak. And you can see that until age about 50 or 55, they run in parallel, and then they diverge, so that this difference increases with age. And that's what that's an index, and I'll explain why in a second, of this increased arterial stiffness. So what I want to do today is give you a brief overview of the two methods that you might not know about, the measurement of cardiorespiratory fitness and the measurement of pulse pressure and arterial elasticity in the brain, which was developed in our lab, and give you an idea of what the study is like, and then give you data about the effect of fitness on a number of variables from brain anatomy to cognition, and also the effects of arterial elasticity in terms of brain anatomy again and cognitive function. So cardiorespiratory fitness can be measured in two ways. The best way, the most objective way, is putting people on a treadmill and measuring basically the output of the utilization of oxygens. This can be done. This is basically what it amounts to, a stress test where you're actually measuring the oxygen consumption. The problem with this is that although this is objective, it gives you a number that is an objective measure, and there are norms, although not very old adult norms for this. The problem is that you need a doctor's permission for this. There has to be a cardiology present for this, and some older adults simply cannot undergo this measure. So if you want to have a study that includes more variability in, say, hypertension or other things, many of those very people may not undergo these tests. So in latest year, Jorka and colleagues have developed what is called the CRF index, which is an index that has a correlation about close to 0.8 with VO2 max, which is the objective measure. And it's based on a number of variables, fitness inventory, the body mass index that was already defined earlier yesterday, heart rate, age, and gender, which are all things that influence VO2 max. And by doing this, it's not only less costly because the stress test costs about $400 per test, but it's also much easier to include people that otherwise would not be able to participate in the studies. So the data we'll present today, a few of them are VO2 max, but the majority of them are done in this second way.
Could you just say two sentences about the fitness inventory?
It asks about things like walking and exercising in an aerobic fashion. So not, if you do yoga or other stretching, it's not count. It's specific types of exercises, and there are parts of this that are fine with older adults, so that they will fit what an older adult, not just the young adults, but an older adult pattern. Now, the norms by Jorka were developed on younger adults, but there are Macaulay and colleagues have developed now norms up to 65, again, age 65 for older adults. Okay, so the research strategy of the study that I'll present today includes, the biggest study that I'll present today includes 55 adults, and they're nicely spaced age 55 to 87. And they are carefully screened. So we only let in people that have had nothing major, which means they're much healthier than probably the average population. They do have some hypertension, but it's not led to any terrible ill effects. They take medications. We exclude anybody who takes anything that affects the central nervous system. So that's pretty much the sample. Now, just like it was discussed yesterday regarding the adolescent participants, again, this is not a random sample of older people. They tend to be, because we have 55, we tend to include people that are probably higher educated than the average and so on. But if you think about aging, what you do by doing this has, in fact, some positive effects. First of all, you are, if anything, underestimating the effects of aging. And second, you are isolating the effects of aging compared to other effects like disease or other things. So you're trying to isolate what the contribution of aging is compared to other factors. So it's not necessarily, it's a bias, but it's not necessarily a bias that changes the data in a way that might give you results that otherwise might not be there. In keeping with the idea that you have to sort of look at multiple factors, we are using a number of different measures that allow us to assess different aspects of physiology and cognitive function. And again, we are looking at individual difference in cardiorespiratory fitness. So why do we worry about all these arterial effects and other effects? And the key is, how do you go from neuronal activity to blood flow, and what's the relationship between them? So many of you, or at least some of you, know that neural activity occurs, but there are consequences, blood flow consequences of it. And here in blue, you will see ways in which these different types of steps between neuronal activity and blood flow changes can be imaged. So you have neural activity, and that is followed by vasodilation in active brain area. This is actually the brain seen in terms of its blood vessels. The brain is highly perfused, so there is a lot of blood, and the brain uses a lot of oxygenated blood. With the vasodilation, there is increased blood flow to active brain area, which is this can be imaged with oxygen 15 PET and also with arterial spin labeling. And then what follows to this is an increase in oxyhemoglobin, because the oxygenated blood that goes to active area flushes out the deoxygenated blood that is there in a manner that is, and more oxygenated blood goes there than it's consumed. This gives rise to the bold fMRI signal that a lot of people use. And also, these two quantities, oxy and deoxyhemoglobin, can be assessed with functional near-infrared spectroscopy. So you can actually have quantitative measures of these two quantities. Now, the catch is that in the young adults, this flow is fairly seamless. But in an older adult, you're going to have to, often you have a reduced dynamic range in this area. Because of hypertension, arteriosclerosis, and plaque, the blood vessel will not dilate the same amount or to the same extent than in younger adults. There is a restricted range. And some measures that we have developed in our lab recently are in fact measures that allow us to assess this stage, the stage of vasodilation, so that we can understand how elastic the blood vessels are. So arterial elasticity is measured in the following way. If you have an elastic blood vessels, you are going to have two waves. During the systole, when the heart pumps out the oxygenated blood toward the brain, you're going to have an increased pressure, which is, however, countermanded by the fact that the arteries are elastic, and so they, in fact, the arterial wall push it back. So there is a sort of a ejected and a counter wave that are sort of in tune. And when the blood vessels are elastic, this lasts for quite a while. This has two advantages. One is that the peak of the pressure is not much. So a peak of the pressure in a blood vessel can lead to rupture or to other, to plaque detachment or what have you, leading to a stroke. So the fact that the pulse pressure is less, it's advantageous, it's a good thing. The duration also allows for an exchange of nutrients that lasts for longer, and so it will maintain the vitality of neurons for longer. So when these two work perfectly, you have this prolonged pulse wave. When the vessels are inelastic, however, these two tend to summate. So you have a very peaky bout of pulse pressure, which is much higher and dangerous for the blood vessels. And then you have nothing, which means that you have less of an opportunity for exchange of perfusion of tissue for nutrients and other things. So this is how this is sort of characterized. But the new things that I'm going to present to you is that you can actually measure it with light. How many of you have seen a pulse oximeter at the hospital, that thing that they put on your finger and estimates your oxygen? So think about the stuff that I'm going to tell you as a giant pulse oximeter that is all over the head, and it's going to give you basically the pulse all over different regions of the brain. So light goes through tissue. This is a wavelength, and this is the absorption spectra of three substances: melanin, water, and oxygenated hemoglobin. There is a window in this wavelength. So this is the visible spectrum. Starting with the red, you're going to have an area where the absorption from various bodily substances is much less, and therefore light can go much deeper into tissue. And I'll give you the 2-minute sort of demo for this. So this is a red laser pointer, and this is a green laser pointer. The green is actually more powerful. If I put my finger on it, you can hardly see anything come out. If I put the red in, you can see that my finger is red. Red light, and more so near infrared light, will go through tissue. The other things that is important for you to see here is that while in air, I can point this laser pointer and see a dot up there. When I put it on my finger, light comes out from all sides. So light is diffused. So we are using this diffuse optical imaging quality to image the pulse, and in fact, it's in other contexts which are not talked about today, to image brain activity. The way it works is that is illustrated in the following picture. So this is an optical pulse, and it's in this case, and in all the data that you'll see, is timed, is synchronized with the R wave from the electrocardiogram, so that we know we are talking about the same pulse all over. So the delays in that pulse, we know, are delays in that specific pulse. What happens is, if a blood vessel is dilated, you're going to have more light being absorbed. So at the peak of the systole, you're going to have the least amount of light. The light goes down. At the peak of the diastole, when there is less absorption of light, so there is a maximum amount. So that gives you the quantity. Now, the normal way that people are used to seeing this is with the systole on top and the diastole at the bottom. So after this one slide, I will show them in this way, upside down, with respect to light amounts, so that you can see them the way that you're expecting to see them. But what I want to show you first is that this is a very large signal. In fact, it's a signal that is treated as an artifact if you're using optical imaging to study brain activity. And in this case, you have this systole, and you can see the signal in single trials. And you can see that there may be difference in young adults, high fit older adults and low fit older adults. And again, as I said, this is a giant pulse oximeter because you can measure it all over the head. We have equipment that can do that. So the last method point, so there are two aspects of this pulse. One is the amplitude, which is bad. The larger, the worse, because it means that your blood, your pulse pressure is high, and high is bad. And then the duration, the longer, the better. So short is bad. So these are the two things. And just to show you, and now these data are inverted to match what people normally expect to see, you see that there is, this is a subject with smaller pulse and a subject with larger. This is the duration. So this is the D wave, which is a good thing, is larger in this subject than in this subject. So you can see that we can, these are individual people that you can see. And again, this would be time-locked to the EKG. So this concludes the methods part of my talk. Yes?
Just your terminology, you say measure all over your head. Are you talking about just the scalp? Or going through part of the bone,
No,
the skull, or what?
We are talking about the cortex. So it's not just the scalp. We are actually using these diffusive methods allow you to go 2-3 centimeters inside the skull. And there are, I can go into the details when we have a discussion. But these are the distance between source and detectors. This is not a direct projection. It's the distance between source and detectors allow you to see basically a volume that is encompassed by banana-shaped kind of flow of photons that are underneath the source and the detector. And the further apart the source and the detector are, the deeper you have. And there are plenty of data on this, which I don't have time to review today. But yes, we are looking at regional pulse measurements in the cortex, not deep inside the thalamus or things like this, but within the external aspects of the cortex.
And you can separate out the effect of the skull from the
Yes.
cortex. Oh,
Yes.
Interesting.
Any other methods question? Because I'm now moving to data, and I want to get through those because that's the main point. The first group of data that I want to show you are data that demonstrate the effects of fitness on brain anatomy, cerebral blood flow, functional brain activation, and cognitive function. So these are relationship between, again, fitness and these other variables. I hope you can see this in the back. These are, again, data from our lab. And this is the unique effect of fitness. So you remove age, education, and gender in a statistical way. And you look at, and these are just older adults, 65 to 85 in age. And you can see that the red area now are areas in which gray matter is more preserved in older adults that are highly fit than in those that are low fit. And this is a study that used VO2 max, the objective measures of fitness. Similarly, and this is a recent sample of 55 to 87-year-old adults, you can see the lower fit, the blue here, is the places in the brain where thinning occurs in lower fit older adults, but it's not there in higher fit older adults. And the interesting part of this is that these are now data from Dickerson's lab at Harvard. And this is a summary of many, many studies done with Alzheimer's disease patients and represents sort of what they call the cortical signature of Alzheimer's disease, those areas that across samples and across studies show thinning consistently in Alzheimer's patients. And the interesting part here is that the lower fit older adults have several areas of thinning that are present in the Alzheimer's patients. On top of that, this area here is one that is considered crucial in terms of thinning to predict which people will convert from mild cognitive impairment to Alzheimer's disease. And you can see these areas being thin in this sample of low-fit older adults, while they are not so in the high-fit older adults. These people are matched for age and many, many other variables. So this is really the difference here.
For the people who aren't good at reading that, would you characterize those key areas?
Yeah, I'm sorry. This is close to the temporoparietal junction, and this is the tip of the temporal lobe here, and you can see more easily here. These are not inflated the same. They're both done with free surfer in terms of software, but they're not inflated the same. Yes?
When you say they're adjusted for many variables, can you say what those are? You said
So,
gender,
yes,
age,
they're
education.
adjusted, so the people have matched for age and gender, and also the vocabulary and education. I mean, these are the kinds of things that you can match for. This is not a huge sample, but we are trying to stratify people
I
that
just
way.
ask because I always wonder with these kind of data, you know, how much we know is cause versus effect. And
Oh, we don't. I mean, absolutely we don't. The hypothesis is that one will cause the other, but these are all correlational data. We don't have intervention, and so you cannot possibly know that. But the link is useful, and I'll show you some mediation analysis that suggests a possible causal link in a second, okay? So what I want to do next is look at a little bit more closely to some of the anatomical data. And now we are talking about frontal lobe regions, and the one that we are looking at is I have tons of these, but I'm just presenting you some sample to give you a sense of what they look like. So it's the pars opercularis and the superior frontal gyrus. And what you see here is data analyzed in two different ways. So this is age in both of the x-axis, and on the y-axis, the normalized brain volume. So this is normalized to intracranial size, which is typical of this data. Now, this is just age regressed onto volume for high fit in red and low fit in blue older adults. And you can see that these two regression lines are the red line is on top of the blue line. On the right, the data analyzed in a slightly different way. So one is now a sample of younger adults, college-age younger adults. So that gives you a sense of how much change has occurred from young adulthood, even though these are cross-sectional data. So they are normalized to 1, which is, again, the younger adults. The other thing is that these are average, so that you take 12 subjects and then you move 1, and it's a running average of 12 subjects, so that you can see more of the trajectory of change, even though it's for illustrative purposes. So there are a number of things that I want you to notice here. The first is that, for example, in this area, there is already about 15% change from young adulthood. The second is that these effects are not huge between high and low fit older adults. They're small effects, but they occur area after area after area, even in a sample this size. So overall, in the end, the pattern is there big times, even though in each single area, the effects are not humongous. In some areas, as I'll show you, the effects are actually large. The other thing, the final thing is that Valerie this morning was talking about pruning. So we don't know, in addition not knowing what the cause and effects are, we also don't know whether this is bad or good. So the assumption is always, more is better. But in this case, some of this pruning may be completely physiological. So if you think of the idea of gist over age, some of it may be useful. I mean, we learn more and more, and we need to keep more and more information in mind. And so the use of gist and the use of and this pruning, so that you can keep things, the pruning might be correlated to this gist acquisition that occurs with age. Of course, there is the point in time where this is no longer good and other things happen. So this is food for thought. We don't have the answers, but these are the data, and it looks like, again, people with higher level of fitness fare better, at least in terms of the comparison with younger adults. This is now the pars opercularis, where, in fact, the effects are steeper, and you can see these effects right here in the other framework. Temporal lobe regions are particularly interesting because when you look at superior temporal, again, the two lines they're always, the red line is always on top of the blue line, suggesting that the high fit have somehow more preserved volume. You can see here that when you normalize to the young adults, you can see that this group holds better than this. But the more interesting data are now the ones for the hippocampus, which is, in fact, a piece of brain that supports long-term memory. You can see here that this is actually an interaction. And what you see here is that this is one. So a lot of the older people who are highly fit, that are 55 or so, still are not very different from the young adults. But there is a drop of this over this period. Of course, this data needs to be taken with a grain of salt, but they're interesting because they match now data with children that show that children who are obese and have low fitness already show differential hippocampus size in infancy, in before pre-adolescence. So many of these, and that's why I'm showing you these figures, might be things that do not occur with aging, that occur over a lifespan, and in fact might give us plenty of opportunity for intervention if we can communicate these kinds of effects to people and engender behavioral change. And again, parietal lobe gives a similar data, so you get the picture. Now, what about brain function? Because I've shown you anatomy so far, and we used in this particular case functional near-infrared spectroscopy, and this is not a very complicated task. This is a response of visual cortex to a checkerboard reversal, just plain sort of brain activation in 3 groups, again, with fitness measured objectively. So young adults, older adults with high levels of VO2 max, and older adults with low levels of fitness. Now, these plots are as follows. You have time in seconds here. This is blood flow, so it's low. And oxy and deoxyhemoglobin concentrations. What you have here is the vertical line here is the moment in which the checkerboard starts reversing. So this is the baseline, and then you have 20 seconds of checkerboard reversal where you have a visual response. What you see here is the young adults and the older adults who have high levels of fitness have a healthy increase in oxyhemoglobin and a decrease. This is graded, but it's not significant in this case. Decrease in the oxyhemoglobin, which is physiological. The older adults who have low levels of fitness don't have much of a response at all. So this is, again, food for thought, both in terms of physiology, what's happening there. You're not receiving enough juice to do the task. And if the task is as simple as this, and if the task is not sustained or fatiguing, you might still cope with this. But if the task becomes sustained and difficult, that might explain some of the drops in performance. And in fact, we have data that I'm not going to show today on the fact that you can have plateauing of sort of activations when you have these situations. The next step is to show cerebral blood flow in gray matter. So this is now measured within the brain with fMRI from a slab that encompasses both some frontal and parietal cortex. And again, what you see here, you have cardiorespiratory fitness and age, and you have the global cerebral blood flow and then frontal and parietal blood flow separated. You can see that CRF predicts or is correlated with cerebral blood flow both globally and separately for parietal and frontal areas. Age is too, but not to the same extent. Age is less correlated with this. So we went and did a mediation analysis to try and see what the relative contribution of age and cardiorespiratory fitness are. So if you regress age and cardiorespiratory fitness, you get some correlation that is significant. But when you introduce CRF, these correlations are bolstered, and the correlation with age goes away, suggesting that CRF is a mediator of this cerebral blood flow effect much more than age is. Again, this is important because it suggests that factors on which we might have control, like cardiorespiratory fitness and the related physiological benefits from it, are going to stave off, or at least are correlated with, in a way, with age in a way that might prevent some of these ill effects of aging. These are also effects of fitness on cognition. So this is a number of tests we did in the neuropsych battery, and these are the effect sizes for high fit versus low fit. Not everything is significant, but again, they're all in the same direction, just like the anatomy. So with the high fit subjects performing better than the low fit subject. So in the remainder, any questions at this point? Yes?
Could you comment on the vocabulary picking up the effect?
In this case, so one possibility of that is that all the fitness and education are correlated, and so that might reflect some of that. But not all of those are, these are either norm for education like CFL or other things, and so backward digispan is not usually correlated with education, yes. The final piece of data that I want to show you is these effects of arterial stiffness, because arterial stiffness we are postulating might be a mediator of this cardiorespiratory fitness effect. And just to remind you, you can remember that this increased distance between diastolic and systolic blood pressure is an indicator of the fact that the arteries are becoming more stiff. And the reason is, as I showed you in the diagram before, that as the arteries become more stiff, the coil and recoil wave are closer, and so there is a much bigger variations in pulse pressure than there is when they are more elastic. So just to show you that the optical measurements work the way you would expect it to work, this is sort of validation. You see age and systolic, diastolic blood pressure. This is our sample. It behaves like the normal situation. So the distance between systolic and diastolic blood pressure increases with age in our sample. If you take the carotid pulse amplitude, which is normally used, and you look at pulse pressure, you also see a big correlation. And in fact, this is the carotid pulse pressure here is measured with optical methods, not with the Doppler. So it's a way of showing that this might work. And this is related to the question about localization and whether this is the brain or not. This is frontal and parietal areas. So this is frontal cortex volume. This is anatomy now. And parietal cortex volume, and this is the frontal pulse and the parietal pulse. You can see that the larger the pulse, which is a bad thing, the lower the volume in frontal cortex and in parietal cortex for the parietal pulse. If you do the opposite, if you look at the parietal pulse and how it predicts the frontal volume, they are not equally predictive. There is, of course, some shared variance. You have a systemic change of stiffness, but the correlations are such that there is some idea that they'll be regionally distributed, that locally in the brain, having a higher pulse in frontal areas will lead to loss of volume in frontal areas and not so much so in parietal areas and vice versa. The final thing, I'm going to present next a couple of physiological sort of functional data. The first is in a task of breath holding. So when you hold your breath, like President Bush nicely demonstrated, lots of stuff happens. So you induce hypercapnia, which is an increase in blood flow CO2, which in turn induces vasodilation and a drop in blood pressure. And that increases cerebral perfusion and delivery of oxyhemoglobin, which you want in order to protect the brain while you're holding your breath. Notice that this task, the breath-holding task, is used as a standardization task because the effects of breath hold are much larger than, say, the cognitive effect that you normally see in fMRI. So by holding your breath, you can kind of gauge the maximum amount of dynamic range of these kinds of effect in the brain. Of course, during a breath-holding task, there is variability, and the more the variability, the more dynamic the system is supposed to be. So variability is good. You want the system to go up quickly and recover quickly. And basically what we did here is using, again, the pulse here was measured optically, and we measure vaso-reactivity across the 4 period. That's the standard deviation across the 4 period. Now, what you can see here that by age, vaso-reactivity goes down, as you was expected to be, except for these 4 people up here. Then you look at cardiorespiratory fitness, and you see that vasoreactivity goes up as expected with cardiorespiratory fitness. Now, these 4 people here, which throw off the correlation with age, are the people with the highest cardiorespiratory fitness, irrespective of their age. So you can see the relationship coming off in this case. And in fact, the mediation analysis, again, on this shows that if you look at age and vaso-reactivity, you have some correlations, but these correlations are bolstered, and the correlation with age goes away when you, in fact, include cardiorespiratory fitness as the mediator. The last two data slides that I have is the use of D-wave. So I've shown you so far the pulse amplitude, this idea that the bigger, the more is this bout of pressure inside the blood vessel, which is not a good thing. In this case, we are looking at the duration, and the longer the duration of this pulse, the D-wave, the better it is. And that's a sign of arterial elasticity. So if you look now at age in this sample and arterial elasticity, you see that the arterial elasticity decreases with age. And the next, this slide that I'm going to show you shows that the D-wave, the duration of the D-wave, is a very good predictor of brain anatomy, meaning that, so D-wave is correlated with CRF, as you would expect it to be. The longer the D-wave, the better, the higher the CRF. But it also predicts, in turn, the total gray matter. I think this is, I can hardly read, the total, this is the subcortical gray volume, so things like the hippocampus and so on, and basal ganglia are included in here, and then the white matter. So the duration of the D-wave is highly correlated with these anatomical variables in the brain. The final slide is the idea that, so what? So you have arterial, stiffness. Does my arterial stiffness today predicts what is going to happen to me next year cognitively? And these are very preliminary data, but the suggestion is that it does. So that these are people now with large pulse, which is bad, and small pulse, which is good. And the pulse is in year 1. We have a year 2 data. This is their neuropsych in year 2 being predicted, or the probability that they'll have a decline in cognitive function measured by neuropsych test battery in year 2 based on the pulse in year 1. So in this case, the pulse in year 1, the neuropsych in year 2 cannot cause the change of the previous year. This is a directional effect. And you can see that, that this is the direction in which this effect takes us. We have lots more data and lots of more analysis to do in this sample. This is, again, as I said, very new data. But basically, just to conclude, I hope I've convinced you that cardiorespiratory fitness has a significant moderating effect on structural, functional, and cognitive changes occurring in aging. And that some of these changes, like some of the anatomical changes, appear to start early in life. And so that would allow ample opportunities for intervention. And intervention data from labs like the Kramer's lab suggests that the beneficial effect can be obtained at any time during the lifespan. And in fact, it doesn't seem to be a really place of no return. People with Alzheimer's disease or Parkinson's disease still seems to benefit somehow from an exercise intervention, suggesting that that's not, an all or known phenomenon. The other point that I made is that the health of the cerebral vascular is an important mechanism underlying the beneficial effects of fitness. And again, both fitness and the cerebral vasculature, which could be influenced also by nutrition, which I haven't talked about at all, are factors that we could intervene upon, that could be trained, that could be changed behaviorally. The cerebral pulse parameter, amplitude, and duration measured regionally from the brain with diffuse optical methods can be used to estimate the status of the cerebrovascular system locally, which is an important point because with appropriate validation, one could hope to predict and, in fact, help prevent cardiovascular accidents that might worsen other things like the onset of dementia and promote healthier aging. And I want to acknowledge a number of people, including Gabriele Gratton, who co-directs the lab with me, and Brad Sutton, who is our collaborator on MR staff, and the NIA for supporting this work. And also, this is the faces. When I say we, I should really say they. These are the they that have done all the work. Thank you for your attention. Yes.
Well, there's a wide range of antihypertensive drugs out there. Some of them will weaken the heart pulse, and some, I think, will relax the muscles inside the arteries. So I'm wondering, to what degree can the arterial stiffness be counteracted by these drugs, and to what degree will this reverse the effects of the...
We have looked a little bit, not formally, because we didn't study. So this is really it was our pilot to see if this was even possible, and it looks like this. So we are now looking for more funding and continuing this work. But the people who take, so there are a few people that take blood pressure medications, a few, like 4, I think, in this sample, that take beta blockers. And what you see is that at least the people who take beta blockers don't seem to have a change in blood pressure. So the correlation between blood pressure, say the difference between systolic and diastolic, goes away, but the stiffness does not necessarily go away with these other measures. So there is, I think that what we should do, and this will be a vehicle for doing these kinds of drug studies, where you can look at the specific effects, the local effects of these drugs on the brain vasculature, which, there is a whole host of things that we should do. So I don't know the answer. There are some dissociations between, taking blood pressure medication and doing that, and having effects that directly reflect on arterial stiffness. And there is also a question of accumulated effect over the lifespan and how much this could be counteracted, say, by a fitness intervention. So the answer is likely to be complicated, and I think in the next wave of data collection that we're going to have, we need to probably allow people with different medication, depending on the ends we have, because the problem is these types of studies require about 5 sessions for each individual subject. So there are 55 people, but they come to the lab about 5 times. You also realize that people who are 87 and can do that are probably genetically very protected from anything. Because even though we try not to have too much of a bias in the sample, the older you get, the more you have survived just fine without having anything. So some of this correlation might break down. Some of this relationship may also not be linear, in the sense that there may be a range, and it looks like that in some cases. There may be a range in which you have really a high risk, where your fitness is below a certain threshold, and above that, it doesn't matter that you are a jock or you simply are healthy and walk around, and you're still gonna, the variance is gonna be explained by other factors. So I'm sorry to make it so complicated, but it's very likely is. Yes. Yes.
So,
Sorry.
so let's say I want to, improve my cognitive function into later life, and I have limited time. Am I better off, I'm thinking of your talk and the talk before, taking the time to be physically active or to do crossword puzzles, Sudoku, and engage in cognitive work?
You'd probably be better off doing both, but I think the physical activity, the data supporting physical activity, in my opinion, are stronger than the data supporting cognitive intervention, in that there are a number of complications. There are a lot of programs now, like apps and so on, that promote this cognitive intervention. And the problem is one of transfer. So you get much better at doing whatever it is that you're doing with these gains, but it's not clear at all that the transfer occurs. There is another complication, and it's one that kind of mars longitudinal study. There is an interesting article by Salthouse talking about that if you do a test year 1 and you do a test in year 2, the same test, these are humans. They're healthy. They remember doing that test before. They have some learning. And so you counteract the effect of you want to look at change, but you're also repeating the same test. And we are kind of trying to work procedures to go around that issue, because the problem is you're seeing some people even improve in year 2 because they've learned, and the people who are doing poorly instead will go down. So this is another issue about this sort of cognitive literature. That doesn't mean to say that being cognitively engaged, have a social life, and so on, by reduce your stress. And in fact, one interesting wrinkle of this data set that, again, I didn't have time to talk about is that there are fairly nice correlations of this kind, the fitness and pulse, with personality factors. Like high neuroticism tend to have bigger pulse, which is bad. And openness to experience tends to correlate with lower pulse and longer D waves. So there are obviously many other factors that can contribute to this. This is just a summary measure that illustrates one aspect of the physiological cycle.
I'm wondering if you could say a bit more about the metabolic elements, because I could imagine that like BMI was part of the indices that got you 0.8% to the gold standard, right? So I'm wondering if, based on what we've heard about how belly fat is worse than other fat, or the relationship between fat and arteries, that maybe it's the triglycerides or certain cholesterols going in there, if you could sort of just magically lyse away all fat, like some fancy liposuction that wouldn't have any of the downsides, would that go a long way toward giving you back fitness, or is it something to do with like muscle mass, things like that?
So we measure girth in every person in this. So we have correlation not only with BMI. BMI is used for the calculation of the CRF index, but we have girth measurements. They do not, the correlation with CRF overall are better than the correlation with girth and are better than the correlation with BMI alone. That's one. Two, that unfortunately there is no magic liposuction. I think that nutrition needs to become part of this equation. Again, this was a 2-year pilot study to look at whether we could even do these types of measurement. I think that nutrition, however, unlike fitness, which is kind of more objective, I mean, you have 4 or 5 things, you can measure them, and you have a number. Nutrition is very complicated. People with different ethnicity will have different nutrition. Macronutrients, which are hardly ever measured in any of these, like lutein, are very important for the brain, even though they are in microscopic amounts. So I think that we can start with diaries, and we can start with some blood assessment of HDL and LDL, but it's not a simple thing to do. I'm sure there are correlations, and in fact, the interesting part is that the genes that support the genes that support aging or vice versa, that are correlated with like dementia or anything that accelerated aging, many of them are also part of the cholesterol cycle. They're part of and so this sort of circle becomes tighter and tighter, uncomfortably so. I mean, I go to the gym every day and I eat low-fat food because of this data. But that's basically the relationship. So it's a complicated one.
We have time for one more question.
Bob.
So one of the interesting things, I think, if I saw it right, was that 2 areas of the brain that were particularly sensitive to the fit, non-fit difference were the hippocampus and the parietal lobes. And so, you're sort of looking at a kind of an Alzheimer's story right away. Mm-hmm. And I was wondering if there's if you have any ideas about, are there special sensitivities of these areas, and I guess I'm mostly thinking about the hippocampus, to vulnerability to these kinds of large pulses and carry out over life?
The hippocampus is certainly hog for oxygen. So if you have anoxia, oftentimes you become amnesic because while your cortex recovers, the hippocampus doesn't have much tolerance for lack of oxygen. So the perfusion of the hippocampus is certainly part of the story. So and again, the data from this, the modeling that I was showing you before really assumes that the cycle of sort of sweeping out the beta amyloid that are there is corrupted when you don't have enough perfusion. So that's one link. For the data for the parietal lobe that I know of are more like empirical in a sense, like this is the data, but we don't know exactly why. The data from Dickerson's lab, for example, show this sort of temporoparietal area as being crucial for the conversion from MCI to the thinning in that area from the conversion from NCI to Alzheimer's. An interesting slide that I haven't presented today because I'm not sure what to do with it is that if you look at delays in blood flow, in when the pulse arrives to an area, that's the last area in the brain where the blood gets. So I don't know what to make of this, but this is an interesting coincidence because just that area is the one where you have a delay, and you can think that if you have a delayed influx of oxygenated blood, systematically, physiologically, because that's where it is. And then on top of that, you might have arterial stiffness so that you have a very punctate bolus of oxygenated blood that arrives instead of a long one. You might think, and that's also an area that we know we have thinning in low-fit people. So if you put all of this together, and I'm really sticking my neck out here, you might think that that's one of the links.
And just for the fun of it, since we're going to stop, one of the other things that, of course, has a huge effect on the peripheral vasculature is this activity of the sympathetic nervous system, which is, God's great vasoconstrictor and dilator. So, and of course, that's intimately involved in emotion, which is one of the things that we're. So, what's your advice as to what kind of an emotional life one should cultivate if one wants to become?
Because I'm the worst possible. I'm Italian, right? So Italians live longer. Actually, so with respect to epidemiology, the Germans live, even though their language is, leads them to savings or whatever, they live 5 years less than Italians. So this is just so that you know.
Probably let those emotions out. Thank you so much.
You're welcome.